Making NAD+ is only half the story. The other half is keeping it from being burned through too quickly. This is where the conversation about diet and brain health gets really interesting, because it turns out that cellular stress is one of the major drivers of NAD+ depletion. When your cells are under oxidative stress or dealing with inflammation, they consume NAD+ at dramatically accelerated rates. This means that protecting your cells from damage might be just as important as providing the building blocks to make more NAD+. Oxidative stress occurs when there's an imbalance between free radicals and antioxidants in your body. Free radicals are unstable molecules that can damage DNA, proteins, and cell membranes. Your cells have repair systems to fix this damage, but many of these repair systems run on NAD+. The enzyme PRP1, for example, is activated by DNA damage and uses enormous amounts of NAD+ to fuel its repair activities. In situations of severe or chronic oxidative stress, PRP1 can actually drain NAD+ reserves to dangerously low levels, leaving cells unable to perform other essential NAD+ dependent functions like energy production. This is where antioxidant-rich foods enter the picture. By reducing oxidative damage, these foods may help preserve NAD+ for its normal roles in energy metabolism rather than forcing the cell to spend it all on emergency repairs. Foods particularly high in antioxidants include berries, dark leafy greens, nuts, dark chocolate, green tea, and colorful vegetables like bell peppers and tomatoes. These foods contain various antioxidant compounds including vitamin C, vitamin E, selenium, and a wide array of plant polyphenol that can neutralize free radicals before they cause damage. The research on this is compelling but not yet definitive. Studies in cell cultures and animal models clearly show that reducing oxidative stress preserves NAD+ levels. We also know from human studies that diets rich in antioxidant foods are associated with better cognitive outcomes and lower risk of neurodegenerative disease. What we don't know for certain is whether NAD+ preservation is the primary mechanism behind these benefits or just one of many protective effects these foods provide. There's also the question of how much protection is actually achievable through diet alone. The brain is particularly vulnerable to oxidative damage because it's metabolically very active, it has high concentrations of oxidizable fats, and it has relatively modest antioxidant defenses compared to some other organs. In Alzheimer's disease specifically, oxidative stress is profound and chronic. Can eating more blueberries and spinach really make a meaningful dent in that level of cellular stress? The honest answer is we don't know for certain, but the biological plausibility is there, and these are foods that offer numerous other health benefits with essentially no downside. One thing to keep in mind is that antioxidants from food work differently than high-dose antioxidant supplements. Multiple large clinical trials have found that high-dose vitamin E or beta-carotene supplements don't reduce disease risk and may even increase mortality in some populations. This appears to be because antioxidants in their natural food matrix work synergistically and are regulated by complex digestive and absorptive processes. When you isolate a single compound and give it in pharmacological doses, you can actually disrupt the delicate balance of oxidative and reductive processes that cells need to function properly. The bottom line on antioxidant foods and NAD+ preservation is cautiously optimistic. These foods likely help, they certainly don't hurt, and they fit into a broader pattern of eating that supports brain health through multiple mechanisms. But we should be careful not to oversell their protective capacity, especially in the context of established disease where oxidative stress is already overwhelming.